So we're done with the ribose sugar, we're done with the nitrogen bases. We have one element left, and that's the phosphate. Okay? Now, phosphate. Is it important? It's probably the most important one so far. Listen, we need to know 4 things about phosphate. Number 1, phosphate forms what is known as the backbone of DNA. Or, well, it's and the sugar form the backbone of the DNA, but without phosphate, you don't get a backbone. I'll explain it, don't worry about it. Number 2, phosphate gives a negative charge to DNA. And this negative charge is going to be extremely important when we talk about DNA organization and how really the DNA itself interacts with something called a histone or histonoctamer. Okay? It's very important. Number 3, we need phosphate for energy, or we use it for energy. Okay? Number 4, and I'll explain all these points, don't worry about it. Right? Number 4. I kind of forgot what number 4 was. Anyway, there is number 4. Just put that in mind. It'll come up. Now, you really want to know what number 4 is? Well, wait a minute. Gotta look at my slides. Oh yeah. Direction. Also extremely important. I'm having a lot of mental blocks today. Direction. What does direction mean? You're going to see this a lot. It's going to be 5' to 3'. Listen, I just want to say and tell you something. Maybe you think we're going into too much detail, right? But believe me, once we reach and we're talking about DNA replication, we're talking about translation, transcription, we are talking about mutation, it's going to be extremely easy for you because you can imagine every step of the process. Eventually, you're going to be so good at biochemistry that any exam you take, any professor that asks you anything is going to think to himself, this guy, he's a fucking legend. Okay? And that's why we're taking so much time. I'm sorry, I know maybe you don't like it, but it's extremely important. Okay, so what do we start with? Let's start with the backbone. Okay? Let's talk about the backbone and how phosphate comes into play into the backbone, right? So, here we go. Now, remember, we said that there's a nitrogen base, and this is a ribose sugar. And since we're talking about DNA here, not RNA, but put in mind, they're going to be made out of the same elements. They're basically made the same way. So at the 2' end, there's going to be a lack of oxygen. At the 3' end, there's going to be a hydroxyl group, right? Now, this and this, the sugar or the nitrogen base that is attached to the sugar is going to be called a nucleoside, right? Now, once we put a phosphate group on it, we call it a nucleotide. And this bad boy right here, now, depends on the nitrogen base, right? So it could be, for instance, okay, let me tell you about the nomenclature really quickly. The 3 things you need to know here. Mono means 1. Di means 2. And tri, it means 3. So if I have 1 phosphate here, I'm going to call it, and this is, well, you could call it a nucleoside by itself and add the phosphate. So basically, you could say that this structure right here, the ribose sugar plus the nitrogen base, which is, a nucleoside, right? And since it has 1 phosphate group on it, we call it nucleoside mono 1. Mono what? Monophosphate. Okay? If it has 2 phosphate groups, what do you call it? Well, it's no longer monophosphate, it's going to be called nucleoside diphosphate, right? If it has 3 phosphates on it, attached to it, Now it's called a nucleoside triphosphate, right? So let's say that this nitrogen base here is adenine, for instance. In this case, this nucleoside, since we put a name to the face, and it has 3 phosphate. Okay, you know what? Let's make it 1 phosphate. Here, nomenclature, right? Let's remove this. So suppose this is going to be the nitrogen base. It has 2 rings. It has an amino group on the 12 o'clock position, right? So it's adenine. So what do we call this? We call this adenine mono, because 1 phosphate, adenine monophosphate, AMP. And you're going to see this. I promise you, you are going to see it a lot. What if it has 2 phosphates on it? Then it's adenosine diphosphate. If it has 3 phosphate, then we call it adenosine triphosphate, the ATP. The energy currency of the cell, right? We always say it's ATP. This is what ATP is. Remember when we talked about the severe combined immunodeficiency, the SCID, right, in immunology, and you said one of the reasons was adenosine deaminase deficiency, right? And you said that there's going to be of course, since this is a DNA, by the way, these all refer to RNA. Why? Well, adenosine monophosphate. If it was DNA, we'd call it deoxyaminomonophosphate, right? Or adenosine diphosphate, or deoxyadenosine triphosphate, right? Now, of course, this would be if it was a DNA, okay? If it's an RNA, then it's going to be AMP, ADP, and ATP. And yes, ATP is a building block for an RNA, okay? Okay, great. So what if it had 2 rings, this nitrogen base, it has 2 rings, and it has no amino group on the carbon number 6. Now what is it? It's a guanosine. And so guanosine, if there's 1 phosphate, it's going to be called guanosine monophosphate. Now, if it lacks an oxygen here, like in this case, it's going to be called simply deoxyguanosine monophosphate, right? And I think you get the idea by now. So if it has 2 phosphates, it's going to be guanosine diphosphate. If it has 3, it's going to be GTP. Again, guanosine diphosphate, phosphate, right? And if it has 3, it's GTP. You're going to see GTP a lot, especially in the signaling when we talk about how the cells signal or intracellular signaling mechanisms, right? So that's the AMP and the GTP. What if it's a pyrimidine? These were purines. What if it's a pyrimidine? So it could be cytosine, right? The nitrogen base could be cytosine. And in fact, if it is, so again, you could say that it's going to be then cytosine monophosphate or cytosine diphosphate, right? If it has 2 phosphate groups, or cytosine triphosphate. Now, of course, you would add if it's part of DNA, if there's missing oxygen here, you're going to say it's deoxycytidine monophosphate, deoxycytidine diphosphate, deoxycytidine triphosphate. If it's uracil, you're going to say uracil monophosphate, uracil diphosphate, and uracil triphosphate. Of course, if the uracil is missing an oxygen group here, it's going to be deoxyuridine monophosphate, right? Don't fall for it. Uracil is never present in DNA. Well, not normally, okay? So you're not going to see it as, you might see it at dump, but not incorporated as deoxy, but it will not be incorporated into DNA. So in fact, this form, you could see it, but it's never going to be incorporated into DNA unless there's an error or like cytosine deamination problem or something like that. Thymidine, the same basic principle. Okay, let's remove this. We kind of, we went off balance. So the backbone, let's continue. Anyway, this is a nucleotide, and now this nucleotide is really, it's ready for building. It comes and let's make an oligonucleotide, let's make polynucleotide, let's make it into a single-stranded DNA, so then it could base pair with another strand and give us a double-stranded DNA. How does that happen? Listen. So this is an active nucleotide, right? Suppose there is another nucleotide here that's carrying another nitrogen base, right? Here's the 5' end, and it has phosphate on it. Now, it actually has to have 3 phosphate groups, but for simplicity, for now, because this is very important. This is, remember we said that we use phosphate for energy. Energy comes from these 2 phosphates, okay? Anyway, for simplicity's sake, for now, let's imagine that this is also, you know, an active nucleotide, right? And here is the 5' end, and of course this is the 3' end. What's going to happen is that this nucleotide is going to come here and stack right here, right? So basically, it comes, this whole nucleotide comes and transfers over here, and of course it uses the phosphate to link the 2 nucleotides with one another, right? So a phosphate here, and here, and now here's a nitrogen base, right? And the 3', here's the 5', there's the 2', remember? And hydroxyl group, right? So what happened here? What happened here? Basically, the phosphate linked these 2 nucleotides with one another. Now, suppose you've got even another nucleotide. Same idea, same concept. So phosphate comes, right? It links another nucleotide with it, right? Here's the hydroxyl group on the 3' end, and here's the 5' end, right? Now, if you want to read this, and this is directionality, by the way, let's remove this 5' end because, you know, it's in the middle, nobody cares about it. Let's remove this because, again, it's in the middle. And so this here, because remember, this carbon in the ribose sugar was the 3', so this would be called the 3' end. The phosphate here, remember, this is a 5' end. And so the way you would read it is, this is the directionality, 5' end to 3' end. Okay, that's all it is. Now, what do we mean when we say the sugar phosphate backbone? Look, what do we have here? It's part of the sugar, right? So the ribose sugar. So here's the sugar. What's this? This is the phosphate, right? Here's a sugar, here's a phosphate, here's a phosphate, here's a sugar. Again, here's a sugar, and here is a phosphate. And so this whole structure, now if we were like to condense it into, you know what, we've got 1, 2, 3, 3 nucleotides, right? And 3 nucleotides, basically, this is the sugar phosphate backbone because it's made out of sugar and phosphate. That's where the weird sugar phosphate backbone comes from. Okay, what about directionality? Well, here's the directionality. 3', 5'. 5' because, you know, is the place where the phosphate attaches to, and 3' here is on the end. If you add another nucleotide here, this would cease to become the 3' now, and in fact, the 3' end would be the end of the incoming nucleotide, right? Great, okay. So why the negative charge on the DNA? What makes the DNA have a negative charge? Well, again, it's going to be phosphate. Now, I could have sworn we had 4 points. What were the 4 points? So we talked about how the phosphate makes the backbone. Oh yeah, listen. So how can I forget such a thing? Energy. Energy, right? Listen. Okay, we need the whole board for this. Sorry, we need it, okay? Before we talk about how phosphate is going to be used as energy to make a DNA molecule, there's a couple of things we have to know. First of all, where does the phosphate come from? Okay, so let's look at the whiteboard here. What's this? This is just a nucleoside, right? Here you've got a nitrogen base, you've got a ribose sugar. Let's make it ribose, for instance, okay? In fact, we're talking about DNA. Let's keep it PJ friendly and make it deoxy, okay? So this is a deoxy nucleoside, right? Here, NS for nucleoside. Let's change the pen, right? And so, how are we going to take okay, before that, what are these? Look at them. So basically, let's look at the sugar first of all. What do we have? We have phosphate, we have sugar, and we have a nitrogen base. Now, the nitrogen base, it's like it has 1 ring and 2 rings, and at the 6th carbon, it has an amino group. And so this means that this is an adenine, right? What are the sugar? Is it ribose sugar or deoxyribose? We look at the 2'. It is what? It is ribose sugar. Great. What about the phosphate? How many phosphate? 1, 2, 3. So 3 phosphate, right? So at this point, we call it adenosine triphosphate. What about the others? Well, the same thing, right? This is also an ATP, and this is also going to be an ATP. Adenosine triphosphate. And so it's a nucleotide, right? That has 3 phosphate groups. Now, this over here is a deoxynucleoside. And so it could be really anything according to what the nitrogen base is. So let's suppose that this is going to be guanosine, right? That this is a guanine, and therefore, of course, it's deoxy, because look at the 2' end, right? There's a missing oxygen group, so it's deoxyguanosine. So how exactly do we take this deoxyguanosine and make it into, deoxy, for instance, deoxyguanosine triphosphate, if it has 3 phosphates, or diphosphate or monophosphate? Well, you put a phosphate on it. If you put 1, it becomes monophosphate, 2 diphosphate, 3 triphosphate. Yeah, okay. Phosphates is not going to be present just freely roaming in the cell. It has to be attached to something. It has to come from somewhere. Now, since we're building a nucleotide, we're going to make a nucleoside. It comes from a source, and that source is the ATP molecule. You're going to hear a lot about the ATP molecule, people. ATP is considered like the currency or the energy currency of the cell, right? Let me tell you something before I go there. Listen, ATP is considered the energy currency of the cell because, triphosphate, because for the mere presence of 1, 2, 3 phosphates on it, right? However, there are other things, there are other currencies to the cell as well, depending on what you're building. For instance, if we want to make proteins, right? We're going to talk about this in translation later on, but let's suppose that you want to build protein. What's the energy currency going to be? It's not going to be ATP. Actually, mostly it's going to be GTP, right? Guanosine triphosphate. And we know that, if you want to make a guanosine triphosphate, just remove the amino group and put an oxygen there, right? And now you have GTP. And so this becomes GTP, not ATP, and you can use it for protein synthesis, right? What else? Yeah, CTP, cytidine triphosphate. Basically, cytidine, again, remember, it's only 1 ring and has an amino group on it, right? What do you use? So GTP, again, for protein. Here we go. Here's protein synthesis. CTP, we use it for phospholipid synthesis. UDP, uridine diphosphate, only 2 phosphate groups, by the way, and we use this in glycolysis, okay? Or I think glycogen synthesis, glycogen synthesis, okay? Now, and there is something else. But let's leave it PJ friendly. We're not going to talk about it here. It's going to take a lot of time that we need, okay? So, the main idea I want to tell you here that yes, ATP is the main currency of the cell. However, it depends on what the cell wants to build. If it's going to make protein synthesis, it's going to use GTP. If it's going to make phospholipid synthesis, we're going to see CTP being thrown around a lot. And we're going to encounter this when we talk about biochemistry. Uridine diphosphate, when you want to make glycogen, right? Okay. So let's remove this. Let's bring back our ATP. So, 1 ring, 2 ring, and here's an amino group. Okay? Again, this is going to be an ATP molecule. Okay, nice. So, we want to make this nucleoside into a nucleotide. And suppose that this is guanine, right? Suppose this is going to be guanine, and so you want to take the deoxyguanosine and make a deoxyguanosine triphosphate. How would you do that? Well, that's simple. You take phosphates and start stacking them. So 3 phosphates, yeah, great. Where do we get the phosphates from? A phosphate, when we are building a nucleotide, is going to be coming from ATP, adenosine triphosphate, through a process called kinase phosphorylation. So what does kinase phosphorylation exactly mean? It's very easy. It's extremely easy. Listen, phosphorylation simply means to take something and put a phosphate on it. So listen, we have Potman here, right? I was gonna, plant something in it and water it and stuff. And now I just use him as an ashtray and as an observer. Anyway, Potman here, if we take a phosphate on it and put it on him, it's gonna, well, we just phosphorylated Potman, right? So we did a phosphorylation process. Okay? Now, Potman's gonna observe us from there. Back to seat, Potman. Yeah. Okay. Good, Potman. Anyway, the cup. Let's say that we want to phosphorylate the cup, okay? Or we put a phosphate on the cup. Now it's called a phosphorylated cup. Great. What about kinase? And remember, when we talked, this is why I tell you to look at the suffix and prefix before every lecture, because everything makes so much more sense, right? What about the kinase? Kinase. So look at the suffix is, that means it's an enzyme. What does the kinase do exactly? The kinase is the hand that takes the phosphate and places it onto something. And according to the target, it's going to be called, for instance, listen, this is guanosine, right? So kinase or guanosine kinase, or in this case, deoxyguanosine kinase. There is an enzyme, and it's called, for instance, deoxyguanosine phosphate, right? Or sorry, deoxyguanosine kinase. What does it do? Well, deoxyguanosine simply refers to this. Kinase is it's going to take a phosphate group. So it's going to take, oh, by the way, okay, we'll go back there. So what's going to do is it's going to take a phosphate group and let it be from this one here, and it's going to insert it at the 5' end of the guanosine. And now we have deoxy, because remember there's no oxygen here, so it's deoxyguanosine monophosphate, right? Which enzyme did the job? The deoxyguanosine kinase. You could say the kinase. It's an easier way to remember it. A kinase is going to take a phosphate and put it on deoxyguanosine phosphate. Here. Deoxyguanosine. And so the enzyme, what's it called? It's called deoxyguanosine kinase. Okay? Great. So, at this point, the deoxyguanosine is now a nucleotide. It's called deoxyguanosine monophosphate. So it has 1 phosphate. Great. Now we need 2 more, right? My desk is too cluttered. Anyway, we need more, right? So what do? Another kinase comes and takes it from this phosphate group and inserts another phosphate right here, right? So now, what's this kinase? What's this kinase called? The one that took the phosphate from this ATP and put it right here. What's it called? It's going to be called deoxyguanosine monophosphate kinase, because it took a phosphate and put it here, so it's a kinase. A kinase to what? What's the target? The target is, well, it's going to be the deoxyguanosine monophosphate, right? So suppose now I've got my second phosphate. What about the third phosphate? The third phosphate, the same idea. We take it from here via an enzyme and we insert it here. So what do you think that, and now of course, we get, when we had 2, they were called deoxyguanosine diphosphate, and when we have 3, it's going to be called deoxyguanosine triphosphate, right? Now, what's the enzyme's name that took that last piece, the third phosphate, and put it right here? It's going to be called deoxyguanosine diphosphate. Why? Well, because at the time, the target only had 2 phosphates, and the target was deoxyguanosine diphosphate. So, a kinase, it depends on the target, right? And by the way, you're going to see the kinase enzyme a lot in biochemistry. I'm going to give you some small examples of this. Okay, listen. Let's remove the phosphate because probably should have left these phosphates. Let's just do them very quickly. Okay, great. Okay, let's remove these pathways and talk about where we could find kinases. It's going to be very brief, I promise you, like 1 minute, okay? Listen, kinases are enzymes that take phosphate and put them on a target thing, okay? So, where will you see it? In metabolism, for instance, glucokinase, right here, glucokinase. People, look at the word, it tells a lot. First of all, is for enzyme. Kinase is an enzyme that puts phosphate on something. What does it put a phosphate on? It puts the phosphate on the glucose, right? So, glucokinase. Okay, what else? Well, yeah, phosphofructokinase. Do not get afraid of the word. It seems scary, it's extremely simple. I promise you that, right? So, phosphofructokinase. This is an enzyme. Enzyme kinase, it's an enzyme that puts a phosphate on something. Where does it put the phosphate? On the phosphofructose. Okay? It takes a phosphate and puts it here. Now, where it gets the phosphate from? Usually from ATP. Could take it from GTP. It depends on the scenario, really. It depends on what you're trying to achieve, right? Because we talked about that. So if you're making protein synthesis, listen, signal transduction, for instance, and we're going to talk about this also. Protein kinase A. So it's a kinase, which means that it takes a phosphate and puts it on a protein called A. Close enough, yes. It phosphorylates a protein. For instance, a very important one is something called the tyrosine receptors, or the tyrosine receptor kinase. So it's a kinase, so it puts a phosphate on the tyrosine receptor. And this is very important, especially when we talk about the insulin receptors, etc. Kinase simply phosphorylates something. And by the way, here's a piece of information that's quite interesting. You know those people that go to the gym and they're like, man, I'm drinking my creatine, just leave me alone, I'm happy, I'm lifting weights. Creatine, by the way, or creatine, according to where we're from, is one of the most studied supplements on God's green earth. It's very safe, and in fact, is very effective. In fact, if you go to the gym, if you're trying to lift weights, trying to increase your muscle strength, the best supplement for you is going to be creatine. Why? Look how creatine works. So, creatine, and yeah, it does have something to do with kinase. So here's a dude, and he's happy. Why is he happy? Well, because he has these deformed Popeye muscles or whatever, right? Here are his muscles. And this dude is muscular because this dude takes creatine. Approximately 5 grams a day should be enough. There's a loading phase. And anyway. And so this dude, because he's drinking creatine, what's happening is that the creatine enters into the muscle cells and then gets transformed into something, right, called creatine phosphate. Okay, and that's nice. So what? So you've got creatine phosphate in your muscles. Well, listen, when we are using our muscles, when we're contracting those muscles, when we're doing, muscular activity or contractions, reps, whatever, right? You're lifting weights, you're using ATP. Now, every time you use ATP, a phosphate, because phosphate is a form of energy, is going to be released, and you end up with adenosine diphosphate, ADP, right? Di. It used to be tri, now it's di. What does phosphocreatine do? Phosphocreatine goes and says, well, not on my watch, ADP, and phosphocreatine, or phosphocreatine, right? Phosphocreatine via the enzyme called phosphocreatine kinase. Remember, kinase, it donates or it accepts a kinase. By the way, the word phosphocreatine kinase here, it's kind of an exception to the rule, because this time the kinase is referring to the thing that's giving the phosphate, which is creatine. It's exception. Okay, anyway, so phosphocreatine kinase goes and gives it gives the ADP a phosphate group, right? And gives it phosphate, and so eventually you replenish your ATP. That's why creatine is such a good supplement. And by the way, creatine is found in beef and stuff. Of course, to get 5 grams of creatine from beef, maybe you'd have to eat 2 pounds a day of beef or something. But anyway, okay, this is how creatine works. Of course, when you have more ATP, it means you can last longer in the gym. You can probably hit heavier weights, and eventually you become stronger, right? And by the way, we have a lecture dedicated to kinase alone. Okay, so that's nice. Let's go back really quickly and talk about how the ATP, what happens exactly, really quickly, right? Why the phosphate, what happens? So anyway, suppose now that this is, adenosine. The nitrogen base is adenine, right? And at this point, what is it? It's deoxyadenine or adenosine. It's deoxyadenosine, right? Because still it's a nucleoside. So what's going to happen is a kinase called, because here's the target, it's going to be called deoxyadenosine kinase, is going to go to this ATP here and take this phosphate group and transfer it on the 5' end. Great. Now, of course, this ATP lost its, before that, very sorry. Just let me tell you something. The way the phosphates are numbered, maybe it's important, maybe it's not. I'm going to do it for completeness sake. Listen, this is the first phosphate, the closest one to the 5' is called alpha. The second is beta, the third is gamma. Okay? So basically, just call them number 1, 2, 3. They could have done that by medicine and everything. We have to make everything so hard. Forget about it. Okay? So alpha, beta, gamma, or 1, 2, 3. I don't care. So, but in textbooks, they say that for instance, deoxyadenosine kinase is going to take the gamma. They could just call it the third one, right? Anyway, they take the gamma phosphate. And so they put it here. And of course, since this is the first phosphate here, we're going to call it the alpha phosphate, or phosphate number 1, right? Okay. What happens to the ATP? Well, it used to be triphosphate, but now it lost, a phosphate here. So what happens to it is no longer called ATP, it's going to be called ADP. Okay, great. So deoxyadenosine, since it has 1 monophosphate, now it's a nucleotide, and we call it deoxyadenosine mono, because 1 phosphate, right? So damn it, you're going to see that. And that's not enough. We need 3, remember? And so again, this is the alpha, beta, and gamma, or 1, 2, 3. Now the deoxyadenosine monophosphate kinase comes and takes this one from here and transfers it here. And now this is called the beta phosphate, right? And so deoxyadenosine monophosphate is, because now it has 2 phosphates on it, is going to be called deoxyadenosine diphosphate, because 2 phosphates, right? Now the last one, of course, the ATP, it becomes ADP, right? And the last one also, alpha, beta, gamma, they take this phosphate and we put it right here. And now, ladies and gentlemen, this becomes, of course, the enzyme that does this is called deoxyadenosine diphosphate kinase. And now we have what we call deoxyadenosine triphosphate, or the DATP, okay? And we saw this, by the way, when we talked about one of the clinical significances of adenosine. Remember, when we said that the severe combined immunodeficiency, I told you there's something called DATP, not to worry about it. I'm going to tell you what it is later. Here it is. It's an adenosine, or an adenine nitrogen base, that's attached to a deoxygenated ribose sugar that has 3 phosphate. We call it deoxyadenosine triphosphate, and accumulates. If it accumulates too much, it causes lymphocytes to die. The kid, he gets sick and failure to thrive and recurrent illnesses, etc., etc. Okay, great. Now this is all very nice, but where does the energy come in? Now, before I wipe this board clean, before I tell you exactly how the nucleotides, now this is a nucleotide triphosphate, how these nucleotides, they stick to one another, let me tell you something. Everything in life needs energy. Now, the more energy you put into something, the better it will be, the stronger it will be. For instance, let's say these are made out of these marker pens, right? They're made out of plastic. And so what I want to do is I want to, fuse them with one another. So I take a torch or, look. I take this, right? And then I start, heating it up. I'll heat this also, okay? Eventually, they're going to start to melt, and I can fuse them with one another, right? Maybe it was, depending on the temperature, depending on the time I use, maybe, takes like 2 minutes, 3 minutes, and then I fuse them with one another. Now, it's plastic, it's fused, yeah. Then I come and I just snap it off. Now, if I left them, if I just left it like this, it probably wouldn't snap on its own, but with a little effort, I can snap it. Now, let's take something metal, for example. What do I have here that is metal? Here, not really. Anyway, suppose these are two pieces of metal, right? And metal is very hard. Metal needs to be, heated to very high degrees, right? So I use a very, very big torch at very high temperatures. Eventually, the metal is going to start to, melt, and now I can fuse these two pieces of metal with one another. I let it cool, and then I come back and I try to break it. Do you think it's going to be easy to break it? No. Why? Because I used so much energy just to fuse them. Now, DNA is the same concept. The same concept is done when we stick nucleotides on top of one another. We need energy, we need a lot of energy because we want it to stay stable. We don't want it, on the first thing that happens, any reaction that goes wrong in the cell to go and just snap the DNA in two, right? That's why we need 3 phosphate groups. Because 2 of those 3 are going to be used to produce energy. Now, let's see how that happens exactly. It's going to be very easy. It's going to be painless. It won't hurt a bit. Don't worry about it. Okay?